Alessandra, are you letting folks in the room now, or?
Yeah.
Okay, Alessandra, you can go ahead and admit everyone. All right, here we are.
I'm just going to wait maybe 1 minute for a few more folks to join. But I will say welcome, everybody. This is the primer on photosynthesis for the Extraterrestrial Photosynthesis Workshop, which is coming soon on May 15th. We have more folks streaming into the room, so I'm just going to wait a moment. Hello, welcome everybody. We'll get started in about 30 seconds to a minute. All right, it looks like the attendance has stabilized a little bit. A few more trickling in, but we have a lot of wonderful material to cover today. So let's go ahead and get started. As I just mentioned, this is the primer on photosynthesis for the Extraterrestrial Photosynthesis Workshop. And the big picture goal of the primer is to provide more information to our astronomer and planetary scientists and other colleagues to give them more background on photosynthesis in advance of the workshop. So at the workshop, we can all move forward together. My name is Nikki Poranto. I'm from NASA Ames Research Center, and I'll be sort of your narrator today in providing transitions between the different information modules of the presentation. Just a note on the code of conduct. Of course, we'll have respectful communication and active listening and some constructive feedback. This will be a harassment-free zone. We're going to be very inclusive. We want to hear all voices from all career stages. And because there will be some presentations of individual research projects, we want to respect the confidentiality and make sure that everything is properly credited and attribution is given. In terms of tech etiquette, we don't want to have any AI note takers, so please don't use those. And then mute your mic when you're not speaking. Yeah, we don't need them. We've got some, when this presentation will be recorded, so you can always go back and reference it. We'll raise our hands for questions, and then you can also post questions in the chat. We have a large and amazing science organizing committee. They span the range of expertise from biophysics to astrophysics and everything in between, and we thank them very much for all of their hard work in helping to organize the workshop and the primers. And special thank you to Nancy Keong and Chris Gisriel and Bob Blankenship for the co-chairs. They put a lot of effort into organizing this. And here are our speakers today. I'm not going to take the time to introduce each one of them, but at the beginning of their sections, they'll introduce themselves and say where they're from and their expertise areas. So with that, I'm going to turn it over to Nancy, who's going to talk about the scope of the workshop, give a recap of the previous primer, and then talk a little bit about other biosignatures that we're familiar with from oxygenic photosynthesis and the planetary scale setting. Nancy?
Hi, thank you, Nikki. Hi, I'm Nancy Keong at the NASA Goddard Institute for Space Studies, where I am an Earth system scientist, biometeorologist, and vegetation modeler. This workshop was put together because the search for life is at a juncture at which we had a decade now and an explosion of discoveries of exoplanets and research about their nature. And also at the same time, there have been very cool discoveries in photosynthesis here on Earth that are stretching our understanding of what possibilities there could be on other planets. So these primers provide background on exoplanet missions and astrobiology to photosynthesis researchers, and also at the same time provide background this primer is to provide background on photosynthesis to astronomers and planetary scientists who have till date been driving a lot of the questions and need the answers from the photosynthesis people. So it's hopefully set a productive meeting in May. And next slide. So last time we had a primer on exoplanet missions and astrobiology The video and slides for these are available on the workshop website, but I'll do a brief recap. Next slide, please. So that primer went over how do we measure properties of an exoplanet, and went over the array of different exoplanet observing space observatories that have been designed to capture different aspects of planets. Next slide. And we also went over what makes the planet habitable. There's great diversity of planets, their stellar parent star to their orbits and formation properties. Next slide. And we also went over how a planet could look different as it co-evolves with the biosphere, looking different in terms of their reflectance spectra that a telescope might see. And this shows how the Archean Earth, early Earth, might have looked with primitive photosynthetic bacteria, the Proterozoic Earth with cyanobacteria and eukaryotes, algae, and then the modern Earth. Very, very different looks and different dominant organisms at different times in Earth kind of histories. Next slide. What this workshop does not focus on solar system planets, although there are photosynthesis questions about life on these, possible life on these planets, but the methods for observing those planets is very different through direct sampling and some. But with exoplanets, we're looking at remote observation with telescopes. So in Mars, people are looking for in situ fossil evidence for life, for example. In Venus, there's an unknown UV absorber in the clouds. Maybe it's light harvesting pigment, probably not, maybe. Titan, people have speculated on possible methane-based photosynthesis pathways. And Europa, there's water in the subsurface, but there could be other kinds of life in photosynthesis. Next slide. Nikki, next slide, please.
Yeah, just a minute. I just lost my the advancement. Sorry about this. The controls for Google Slides just disappeared. I'm going to have to stop and reload this again.
Sorry about that, everyone. Thank you, everyone, again for joining the call. We have a lot in store today, and the talks today will be a little bit more extensive. So thank you for extending your time about 15 minutes longer for this primer. Okay. Sometimes you have to clear your cache.
Yeah, I deeply apologize. I don't know what's going on here. Mm-hmm. Everything I try, yeah, I'm trying my keyboard buttons as well. Hmm.
Huh. Okay, you know what I'll do is I'll share the slides. How's that? Okay. Let me do that. All right. Okay.
It's still not advancing for me.
Okay. I'll share the slides right now. Let's see. Where's the sharing slide? Oh, you're sharing a slide now, but you can't advance?
No, I can't advance.
Okay, all right. Oops, that's not. All right. Can everyone see that fine? Okay, yep. And I hope there's not a terrible hum. Okay, so I'll advance the slides then. So this primer on photosynthesis is going to cover a few things. For one thing, don't read all these questions now, but when you come out of this primer, we're hoping you will be able to read these questions and understand why they're being asked, and we can revisit these later. But some of the questions include, like, what are the limits to the number of photosystems that can be strung in series to support electron transfers? And why is that important for astrobiology? Okay. The outline for the rest of the presenters is that we'll go over some planetary scale biosignatures, drill down to the molecular level of what is photosynthesis, and go over the diversity of colors of photosynthesis, possible alternative ways photosynthesis might be configured on other planets, and then we'll have our group activity. Why are we looking for photosynthetic organisms on other planets? Well, it's because we know that on our planet, photosynthesis is a major life process that produces two very strong signs of life that we can see from space. On our planet's surface, there's something called the vegetation red edge, which you heard about in the first primer. And there's also atmospheric oxygen. On the left is a plot showing the solar spectral irradiance from our sun to the top of our atmosphere, and the transmission of the light through our atmosphere to the surface, what organisms on the surface actually get. And the green line is the reflectance spectrum of a plant leaf, in this case, a grass, which is very representative, although there can be variation. So this shows kind of a low reflectance in the visible, because photosynthesis, oxygenic photosynthesis uses a lot of visible light. There's a green bump where there's a little less absorbance of light, but still quite a lot. So our eye sees the green. And then from the red to the near infrared, there is this steep contrast with a rise in reflectance in the near infrared. And that step-like feature is known as the vegetation red edge. It is so distinct from minerals on our surface, or any dominant minerals anyway, that satellites use this to track the presence of vegetation over the surface of our planet. And why does this leaf have this reflectance spectrum? Well, one is because of chlorophyll A being the dominant photopigment. And this green line now here shows the absorbance spectrum, not the reflectance spectrum of chlorophyll A. It absorbs strongly in the blue and in the red. And then it cuts out a little bit tail off in the far red. The near infrared reflectance is because of the leaf structure. In very healthy turgid leaves, the healthy cell walls have a change of index of refraction between the cell wall and air spaces in the leaf. If the leaf wilts, or even in lichens, that red edge is much weaker. You have much less scattering in the near infrared. So how is that expressed at the planetary scale? These show plots of the reflectance spectrum of our Earth as seen in the Earthshine. And this is a nice class of studies to go look up. The Earthshine is the reflectance of the Earth off the moon, which is a nice disintegrated average of our planet's reflectance as might be seen by a telescope. And you can see the vegetation red edge can show up as a bump if there's a continent is in view, and it varies a bit depending on which view of the Earth you're getting. This reflectance spectrum also shows oxygen in our atmosphere. And oxygen is important because it's very hard for a dead planet to build up oxygen in its atmosphere. There's false positive cases, but largely it's a strong sign of life, because otherwise oxygen would be rained out by water, or it would oxidize the crust. Now, oxygenic photosynthesis is not the only kind. There's also anoxygenic photosynthesis, and our later speakers will get much more into detail about these. But these kind of organisms have light harvesting pigments, but they do not produce oxygen. And different kinds of photosynthetic organisms dominated at different times in our Earth's history. Here's a diagram of the evolution of atmospheric oxygen on our Earth. Early Earth did not have oxygen. It had a reducing atmosphere. And the earliest life, maybe 3.6, 3.5 billion years, were archaea and anoxygenic phototrophs, like the green, purple bacteria and green sulfur and purple bacteria you saw on the previous slide. And they didn't produce oxygen. And later cyanobacteria that could produce oxygen arose, and it took a while to oxygenate the atmosphere. And there are little whiffs that you can see of oxygen in the fossil record. And finally, we had the great oxidation event around 2.4 billion years ago, when there was a steady rise now in atmospheric oxygen, and that it kind of was at low levels for a very, very long time, when there were mostly just cyanobacteria and algae in the oceans producing oxygen. And then finally, plants emerged onto land, and and the oxygen stabilized close to present day. Now those plants continue to evolve just to spread across the land and evolve structures but the photosynthetic apparatus was largely preserved. They largely evolved structures to gain carbon, to gain light, to gain water, and to promote efficiency and spreading in the environment and greater productivity. But the photosynthetic apparatus is fairly well preserved, but we'll get into that more with later speakers. And the Earth is still evolving, and possibly some of these later pigments that have been discovered recently, maybe they will take prevalence or not. Jury's still out, but maybe for sure Earth, maybe the red edge will change color. Who knows? So this plot we saw before about how the planet changes in its look to like a space telescope over time. The early Archean Earth, the Proterozoic Earth, and modern Earth, all different spectra with different organisms dominating at different times. So that's the planetary scale, but we want to really dig down into the molecular scale and other scales along the way that affect what we finally see in the signal from a telescope. So of course, a planet has diverse climates and therefore diverse ecosystems and different morphologies of organism. In this case, I'm just going to go over oxygenic photosynthesis. For plants, the basic unit that creates a spectra of interest are the leaf level, where you have gas exchange and the photosynthetic apparatus, and you have the structure of the leaf that drives a lot of that vegetation red edge and the near-infrared reflectance. In those leaves, the cells contain what are called chloroplasts, and these are the organelles that contain the machinery for photosynthesis. Inside these chloroplasts are stacks of membranes called thylakoid membranes, and here's the electron micrograph of what these actually look like. And these are these membranes also I'll show blow up later. The chloroplasts only exist in eukaryotes, but they existed because of endosymbiosis of a cyanobacterium with another protist. So cyanobacteria kind of like their own chloroplasts, and their thylakoid membranes can have a different architectural arrangement inside for interception of light. So this is a cartoon blow-up of the thylakoid membrane, and you can see it's a lipid bilayer on which the various molecules sit for the machinery of photosynthesis. This includes a chlorophyll A, which is the main photopigment. There are also antenna pigments that allow a broader absorbance spectrum across for solar radiation. And then there's the oxygen evolving complex, which is the critical molecule for splitting the water. And this is a unique molecule in nature. And Bob and Chris will go into that a bit more. And of course, then all this cruises ATP to provide energy for the organism. So next, back to you, Nikki.
Great, thank you. Apologies for my glitch earlier. I'm on a NASA computer and we don't typically use Google Meets. So next we're going to hear from Bob Blankenship. He's going to tell us what photosynthesis is. Bob?
Hi, my name is Bob Blankenship. I'm a retired biochemist. And I'll first start off with a kind of a textbook definition of photosynthesis. It's a process in which light energy is captured and stored as chemical energy by a living organism. So that distinguishes it from purely a chemical photochemistry type thing, situation. The stored energy is used to drive energy requiring cellular processes. So this is a general definition, covers a variety of different types of photosynthesis. Next slide. So on Earth, we have two main types of photosynthesis. One we call chlorophyll-based photosynthesis, which is the one we're all familiar with. And that includes the sorts of complexes that Nancy showed on the earlier slide. And that's found in bacteria and also all the eukaryotic photosynthetic organisms, fixes CO2. Most of these organisms are what we call autotrophic. But there's another form of photosynthesis called rhodopsin-based photosynthesis, and it relies not on chlorophyll, but on retinal type pigments, similar to what you have in your eyes. And it works by doing cis-trans isomerization and proton pumping or ion pumping. And this is found in certain types of archaea and bacteria, usually uses organic carbon, and these organisms are not autotrophs, they're heterotrophs. So next slide. We'll focus mostly on chlorophyll-based photosynthesis. And here we have a distinction we need to make. First, that the photosynthesis is intrinsically a redox process. That means reduction oxidation. Electrons are being transferred from an electron donor to an electron acceptor. And the electron acceptor in most of these cases is carbon dioxide. So we distinguish anoxygenic and oxygenic photosynthesis. And here the anoxygenic is represented by having the electron donor be hydrogen sulfide, although other things can serve. This is perhaps the most common. And so the sulfur is oxidized to elemental sulfur, and the CO2 is reduced to CH2O, which represents carbohydrate. In oxygenic photosynthesis, the sulfur is replaced by oxygen, and the product is O2, molecular oxygen, which is what accumulates in the atmosphere and has transformed the Earth. So next slide. If we look at the tree of life, and this is a tree of life based on 16S RNA sequences, and the colored ovals are all the various types of photosynthetic organisms, we can see that most of these are present in the what we call the bacterial domain, the 3 domains of life. And then there are a number of photosynthetic organisms, of course, that are in the eukaryotic domain, algae and plants and so on. And all of those have arisen through a process called endosymbiosis, in which a cyanobacterium was incorporated into a proto-eukaryotic cell and became the cell organelle, the chloroplast. Now, the different colors there represent whether we're talking about one type or another of the reaction center complexes that are in these organisms, and we'll talk about that in more detail later. One of the interesting things to note is that there's a complete lack of any chlorophyll-based photosynthesis in the archaeal domain. And so as far as we know, there have never been any archaea that do chlorophyll-based photosynthesis, although many of them do the rhodopsin-based photosynthesis that we mentioned earlier. Next slide. One of the basic concepts that apply to all chlorophyll-based photosynthetic organisms is the idea of light harvesting and energy storage through the reaction center complex. Most of the pigments are organized as a light harvesting antenna, and so sunlight is a relatively dilute energy source, and so a number of pigments can absorb a photon, and then through a process of energy transfer, that energy is delivered to a special complex called the reaction center, and that's where the electron transfer process that actually stores the energy in a chemically more stable form. And you can think about this a little bit like a radio telescope or satellite dish, with the antenna being like the dish and the reaction center being like the receiver that transduces the signal. All different chlorophyll-based photosynthetic organisms contain a light gathering antenna system. It has a lot of variation, as we'll see in a minute. And the reaction centers, on the other hand, are much more conservative, and they seem to have derived from a single ancestral form, and all of the reaction centers we found come from a single invention, whereas the antennas have been invented multiple times during evolution. Next slide. This gallery just gives us a little sense of the diversity of these antenna complexes. These primarily serve to adapt the organisms that contain the different types of antennas to very different photic environments. For example, in the upper left, the chlorosome antennas that are found in the green sulfur bacteria, they adapt these organisms to extremely low light intensity regimes. And you can do a back of the envelope calculation that each of the chlorophyll molecules might absorb a photon once every 8 hours. So these things are on the very edge of not having enough light to work. Another example in the lower left is the pyridinin chlorophyll complex, which is found in dinoflagellate algae, and that is primarily has pigments of carotenoids, which absorb in that middle green region, and this suits these organisms to that photic environment. They're down in the water column, and at that point, it's primarily the blue-green light that penetrates down that deep, and so they're well suited to life there. And you can make similar stories about these various other types of antenna complexes. And so they're really remarkably diverse and clearly have resulted from multiple evolutionary inventions. Next slide. This is a cartoon that shows the energetics that happen in the reaction center complex. We're not going to focus on that aspect of it. And the most important thing there is that the pigment at the core of the reaction center complex will absorb a photon, usually by energy transfer from the antenna, and that then promotes it to an excited state, which is represented by P star in this diagram. And that excited state has a rather loosely bound electron, which can be an extremely strong reducing agent. And that then is quickly lost to an acceptor, and then there's an electron transfer chain that carries it and finally to a terminal acceptor. And on the other side, the oxidized P is reduced by an electron donor. Ultimately, in oxygenic photosynthesis, this is water that is the donor. And these primary reaction is the absorption and the oxidation of the pigment P, and then the secondary reactions stabilize that primary photo product. Excuse me. Next slide. We can take a broader scope of these reaction center complexes, and in the middle is the famous Z scheme of oxygenic photosynthesis, in which there are two photosystems, each of which have that photon absorption and excited state electron transfer. They're coupled together in series by a electron transport chain, but there are also a variety of anoxygenic bacteria, primarily that work with cyclic electron flow, and those are shown on the left and the right, and they are found in the various types of anoxygenic bacteria. But this shows a sort of unity of the different types of reaction centers, and it's not shown here, but they do ultimately come back to what we think is a single evolutionary source. Next slide. Finally, we'll just look at the carbon fixation part of the process. This is complicated cycles here. The one that's probably most familiar to people is the Calvin cycle. Excuse me. Which uses the carboxylating enzyme rubisco, ribulose bisphosphate carboxylase oxygenase. That's found in cyanobacteria and algae, but also in some purple bacteria and other organisms. And so that's the main way that carbon is fixed, and we won't go through the details of that complex cycle. But there are other alternatives in some of the anoxygenic bacteria. There's what's called the reverse tricarboxylic acid, or RTCA cycle, which is found in green sulfur bacteria and some others. And then the hydroxypropionate cycle, which is found in the chloroflexus type bacteria and some others as well. So these are different ways to skin a cat in terms of fixing carbon dioxide into carbohydrate. Next slide. So I think this is where Chris takes over.
Yeah. Hey everyone, I'm Chris Gaisrael. I'm an assistant professor at the University of Wisconsin-Madison. I am also a biochemist and most notably a structural biologist as well. And hopefully I'll be able to put some of what's been described into even more of a sort of physical context. So in this slide, I'm just kind of showing that you can think about photosynthesis on different scales, right? The sort of planetary scale on the top left, the scale that you and I are all most familiar with, that's on our scale, thinking about, trees and grass outside, that sort of thing. Down from there, so we're now talking about 10 to the minus 7 meters, we think about the membranes, photosynthetic membranes that contain the pigment protein complexes that are involved in photosynthesis that have been being discussed. And then we can think about an even smaller scale, which I think some people are not so used to thinking about, where what we're showing are different amino acids. So this is the protein and how those amino acids bind different cofactors. And I would call cofactors small molecules that are bound by the protein, and their purpose is to serve some sort of functional purpose, right? Some sort of catalytic mechanism. So obviously, these are all tightly connected despite spanning 17 orders of magnitude. So next slide. And again, for those of you who are unfamiliar with looking at pictures like this, what I'm showing you here, this is a picture of one of the two photosystems of oxygenic photosynthesis. This one's called photosystem 1. You don't need to worry about exactly which one, but I've kind of grayed out the contribution of the protein and a lot of the cofactors. And the cofactors that I am showing are the chlorophyll molecules. So chlorophyll, as was said, are the primary pigment or chromophore that are involved in at least oxygenic photosynthesis. The structure of chlorophyll is on the right. It has this ring type structure where this double bond, single bond sort of conjugated system that results in the ability for this pigment to absorb visible light. Next slide. And then now I would like you to recall the description that Bob made, where you have this energy transfer. So look at the top right. You have energy transfer from these different antenna complexes, and that energy goes to the reaction center where there's electron transfer. Well, we can put that into more of a physical context in the image that I'm showing in the center, where you have all of these chlorophyll molecules, and maybe you have a photon being absorbed by one of these chlorophylls. Excitation energy transfer occurs where that energy is moving through a number of different pigments, usually chlorophyll molecules, until it gets to a certain place. And this is kind of where the magic happens of converting the energy of light to chemical potential energy, right? This is really the input of energy into our biosphere, period, right? And because of that, you have electrons move from one side of the membrane to the other, but importantly, on sort of the bottom of the image shown here, that electron does not have the reducing power to be able to drive metabolism, but by coupling, by essentially increasing its energy as a result of using that from light, it now has more energy so that it can drive those downstream metabolic actions. Next slide. And then I just wanted to make a comment about photosystem 2 and water oxidation here. I'm showing the structure of photosystem 2 on the left. You don't need to worry a huge amount about that, but basically where I'm showing the little stars, if you look closely there, you can see where the electron transfer cofactors of photosystem 2 are. And they comprise a number of different molecules, but the important one I want to show you is this one that I've colored in sort of purple and red and green there. That's a metallocofactor that people often refer to as the oxygen evolving complex or the water oxidation complex. I've zoomed in on the top right to that, and in the center of that circle, you kind of see that it's made up of manganese ions and a calcium ion that are connected by these oxygen atoms. They call those mu-oxo bridges. You can see that there's amino acids on the outside of coordinating that. And then on the outside of that big circle is this kind of multi-step catalytic mechanism that is used to describe the water oxidation process. So as was described before, the source of electrons for photosystem 2 and really for photosynthesis in general is water, right? So that water splitting reaction by this cofactor is what ends up fueling metabolism, but also releasing the oxygen that you and I know and love, and that leads to all higher life on Earth. There's relevance, of course, to this workshop. If an important biosignature is oxygen on other planets, we have to ask, how did this evolve on Earth? And then is it plausible for it to arise on another planet? Next slide. All right. Do you want to say something, Nikki?
Yeah, I'll just transition us. So now we're moving into talking about the colors of photosynthesis. So Chris is going to address the diversity of photosynthetic pigments, their protein environment that you've heard him mention just briefly, and then next, the far red limit of photosynthesis.
Okay, so this is a slide that Nancy showed before, and I just want to drive home a couple important points here. So on the left-hand side, we're looking at oxygenic photosynthesis. We talk about plants and algae, we talk about cyanobacteria. I have to stress there's much more sort of diversity among cyanobacteria than there are plants and algae, and that's a really important thing to study, right? Where they can thrive in different light niches and stuff like that. On the right-hand side, we see a couple of anoxygenic phototrophs. On the top right, you see purple bacteria. On the bottom right is an example of a habitat for green sulfur bacteria. This is actually on the bottom right, at the bottom of the ocean around hydrothermal vents, where these bacteria are growing not off of light from the sun, but actually off of light from the heat generated from the center of the Earth. So, in fact, one of our co-organizers, Bob Blankenship, took a submarine down to a place like this and was able to sample some of these bacteria. But an important point I want to mention is that there is much, much more diversity among anoxygenic phototrophs than oxygenic phototrophs. And you'll see what I mean by that in just a moment. Next slide. Okay, thinking more about this sort of molecular scale of photosynthesis, we already introduced these chlorophyll molecules on the top left, and Nancy had mentioned chlorophyll A. Well, there's a number of different types of chlorophylls, and the chlorophylls differ by the chemical substituents that are on the outside of that main ring. Don't worry about exactly what they are, but the point is you can make alterations to that chemical and see different shifting in the type of light that's absorbed. You can also look at the same thing for carotenoids, so hopefully you can see the absorbance spectrum on the bottom dependent upon changes in that molecule. Same with bilins. These are antenna pigments that are found in cyanobacteria. But an important point is that it's dependent, that this absorption is dependent not only upon the chemical species that's bound by the protein, but by the protein environment itself, right? The amino acid interactions around it, that sort of thing. Next slide. All right, and we can see an example of one of those things right here. So you have chlorophylls and bacterial chlorophylls. Chlorophylls are found in oxygenic photosynthesis. Bacterial chlorophylls are found in anoxygenic photosynthesis. You don't need to worry about the chemical difference, but an important point is kind of shown in the absorbance spectrum on the bottom of this view. If you look at the top, the black line is just the light irradiation spectrum of the sun that makes its way all the way to Earth's surface, right? So lots of photons, especially in this visible region of the solar spectrum from 400 to 800, or 700 nanometers. And you can see that the chlorophylls that are involved in oxygenic photosynthesis, so especially look at chlorophyll A, that's the green line, it's absorbing light that's higher energy or lower wavelength than around 700 nanometers. And that's kind of what people used to think is a strict cutoff. In anoxygenic photosynthesis, where you see these bacterial chlorophylls, you see it's much more redshifted, right? It requires lower energy to obtain those reducing equivalents to drive metabolism. In fact, there's some pigments in anoxygenic photosynthesis that absorb light all the way out to 1,000 nanometers. So it's something to consider when we think about what life could look like on other planets and different biosignatures. Next slide. And then finally, I think this is the last slide, this idea of a red limit for oxygenic photosynthesis. So I'm kind of showing one of the most extreme case that I know of here. There's a process in cyanobacteria called far red light photoacclimation. We often abbreviate it FARLIP. And what happens is, whereas certain cyanobacteria typically only use chlorophyll A in these photosystems, in some cases, they can switch out some of those chlorophyll A molecules for chlorophyll D and F molecules, and those are redshifted relative to chlorophyll A. And if you look on the right-hand side, I've just put little arrows where you can see the contribution of those redshifted chlorophyll molecules to the absorbance spectrum of the photosystems there. And the main point is these guys can use 800 nanometer wavelength light in order to drive oxygenic photosynthesis, or in other words, water splitting at photosystem 2. So right now, the current sort of limit of how we understand the red limit or low energy limit of oxygenic photosynthesis is about 800 nanometers for now. I think that's all I have. Yeah.
Thank you. So next we're going to hear from Nicoletta LaRocca, who's going to talk about tuning chlorophyll A, and then also about different architectural strategies that higher organisms can use to adapt to low light and far red light conditions.
Okay, thank you, Nikki. And I'm Nicoletta LaRocca. I'm associate professor at the Department of Biology, University of Padua in Italy, and I'm a microbiologist and plant ecophysiologist. And starting from that Chris said, organisms can work also with longer wavelengths and so low energy photons. And this is true for cyanobacteria that synthesize new pigments such as the chlorophyll D and F that absorb at longer wavelengths. But also a number of microalgae and plants have been found and been discovered, and we hope to discover other of these organisms capable to use infrared light, the only infrared light for oxygenic photosynthesis. And they do that just using the canonical chlorophyll A and changing the proteic environment that harbors this chlorophyll A. And thanks to the proteins environment, the chlorophyll A shifts its absorption to longer wavelengths. Next click. Okay, and in that way, the molecules of chlorophyll can be coupled and enhance their excitonic coupling and be able to utilize also low energy photons in the infrared. And beside that, please go on. Okay. Beside that, the organisms that have been discovered to do that, eukaryotic organisms, are also utilizing different architectural and structural strategies to enhance the light harvesting in low, very low light and infrared light. Please go on. We have understory plants that experience a light spectrum, such as that you can observe in the left upper part of the picture. So very dim in the visible light and rich in infrared. This type of plant has structural adaptations such as conic epidermal cell that can intercept light coming from different directions and focus this light towards the photosynthetic layer of tissue. And in this tissue, we have giant chloroplasts, so very big chloroplasts with membranes that can be stacked in superstacks or ordered in very ordered way. And this has a nanophotonic effect that enhances the use of very low and infrared light. Gone. Also, some algae that live in shaded niches are able to utilize even just infrared light, and they also show changes in cell morphology and also giant chloroplasts with superstacks. These superstacks are thylakoid membranes that can have nanophotonic effects, functioning as efficient nanoscatters and leading to the photon propagation. And this significantly enhanced the photosynthetic light harvesting, particularly in low light and infrared light.
Great, thank you. So you've heard about the diversity of pigments, you've heard how pigments can be tuned. Now we're going to change gears slightly. You're going to hear about even more possibilities in what artificial photosynthesis can reveal. So next we'll have Gary Moore.
Thank you, Nikki. I'm Gary Moore from Arizona State University School of Molecular Sciences and the Biodesign Institute Center for Applied Structural Discovery. Today I'll share with you ideas on artificial photosynthesis, human-engineered technologies that are emerging that convert sunlight to chemical power or electrical power. The slide shown now is one that's been collected data since the mid-70s by the National Renewable Energy Lab, now the National Laboratory of the Rockies. And this slide highlights how there's various materials and approaches that can be used for converting sunlight to electrical power. And these have a wide range in efficiencies they can achieve, upwards of 50%. These materials use different fabrication processes, different types of light-absorbing materials. And this 50% value is one that stands out as it's above a concept that comes out of solid-state physics called the Shockley-Quasar efficiency analysis. This analysis shows that for a material that absorbs a single wavelength of light as a junction band gap, about 34% is the most efficient you should achieve. And that material would have a band gap of about 1.34 electron volts. These materials that have higher efficiencies don't violate this principle, but they use tandem architectures, multiple junctions. Next slide, please. Now, converting sunlight to electrical power in terms of photosynthesis is not the complete picture. Photosynthetic organisms convert light into chemical power. We saw how they can use water as a source of electrons to produce reducing equivalents and energy carriers. But this machinery evolved under a reducing conditions in Earth's atmosphere, where the electron sources were not quite as capable of oxygen evolution. And photosynthetic organisms, the machinery had to evolve to span a relatively large redox span for carrying out water oxidation chemistry. This raises questions of what photosynthesis could look like on other planets where the chemistry of the electron donors may be different. Next slide, please. Of course, in artificial photosynthesis, it's not every aspect of biology that's a target of chemical mimicry, but rather just the specific reactions that are potentially useful to humans. In a way, the artificial photosynthesis gives us not only the ability to think about what biological photosynthesis might be like on other planets, but could we look for technological signatures as well, where advanced civilizations are developing technologies to use solar energy for powering their economies. Next slide, please. This slide highlights an example of an existing form of artificial photosynthesis that's being compared with the natural biological process of photosynthesis. Using existing technologies available today, you can, for example, wire 3 photovoltaics in series. This allows the voltages to stack in a way to meet that thermodynamic demand of 1.2 volts. We're using water as a source of electrons, generating a current, and storing those electrons in the form of chemical power as hydrogen. In natural photosynthesis, this also uses water as a source of electrons, producing a thermodynamic equivalent of hydrogen, NADPH, and generating what you might call a wireless current. In addition to generating electricity, plants also make use of proticity, that is by pumping protons and generating proton gradients across membranes. Next slide, please. In artificial photosynthesis to create fuels, there's also a wide range of approaches that are being explored. I highlighted one on the previous slide that has what you might call a high technology readiness level. This integrates a separate light absorbing unit with catalyst for carrying out the water oxidation and fuel forming processes. But researchers across the globe are looking at materials that are more integrated, ones where the light harvesting material also carries out chemical processes. Next slide, please. In all of these approaches, despite their varying ways that they try to achieve photosynthesis or artificial photosynthesis, one thing that stands out is a challenge that's sometimes been referred to as a golden triangle. And that is that for practical fuel production, you would desire to have an efficient system. It should also be durable and be built from materials that are scalable or earth abundant and thus low cost. But achieving this has been a challenge. Most approaches can achieve 2 of the 3, and you trade off between these. I think that's the end of the presentation, and I'll turn it over.
Great, thank you, Gary. So next we're going to hear from Nate Ennis. And so you've heard Bob Blankenship mention this Z-scheme of photosynthesis. So he's going to talk about rewiring the Z-scheme. Nate?
Thanks, Nikki. Yeah, I'm Nate Ennis. I'm an assistant professor of biochemistry and physics at North Carolina State. So we heard in this talk about oxygenic photosynthesis being the dominant form of photosynthesis on Earth. There's also anoxygenic photosynthesis, which we also heard about. So in anoxygenic photosynthesis, you can use near-infrared light, but you can't oxidize water. So here we're taking a closer look at the Z-scheme of oxygenic photosynthesis. So oxygenic photosynthesis is responsible for the red edge and the green bump in Earth's spectrum. And so water oxidation, which happens in the lower left of this diagram, is why oxygenic phototrophs were able to proliferate. So So in this Z scheme, we've got energy or redox potential on the y-axis. We've got photosystem 2 on the left, photosystem 1 on the right. And so we're tracing the trajectory of an electron through the electron transport chain. So it starts out in the lower left. P680 oxidizes tyrosine, which oxidizes the oxygen evolving cluster, which oxidizes water. So that lower blue dashed line, that's the redox potential at which water oxidation occurs. So PS2, its big job is to take electrons out of water. So it needs to be oxidizing enough to do that. And then on the right-hand side of the scheme, we've got photosystem 1, which needs to be reducing enough to make hydrogen equivalents, or NADPH. So it can reduce ferredoxin, which eventually leads to NADPH and then carbon fixation. So oxygenic photosynthesis has the advantage of being able to use light for water oxidation, which allows it to proliferate and live anywhere where there's water on Earth. But it has some limitations. So one is that it can't access the near infrared. Another is that PS1 and PS2 compete for photons in the same wavelength ranges. And a third one is that for every electron that comes out of a water molecule, you need 2 photons to be absorbed, one in PS2 and one in PS1. So on the next slide, we've got a hypothetical photosystem where instead of using red photon energy, we're using the energy equivalent to a green photon. So you can imagine a flavin or a porphyrin or some other molecule that has its lowest energy excited state in the green, that drives water oxidation on one side of the photosystem and makes hydrogen equivalents, or NADPH, on the other side. So in this case, we'd be using half as many photons to do both half reactions of photosynthesis. The drawback, though, is that we wouldn't be able to use red light or near-infrared light, so we're more restricted in the photons that we have access to. So on the next slide, we could imagine a system where we're mixing different types of photosynthesis together. So in the upper left is the native Z scheme that is limited to the visible range. We've also, so there's some hypothetical water oxidation pathways that use near-infrared light in the upper right and lower left. And then in the bottom right is a high-energy photosystem that uses high-energy photons more efficiently than the other photosystems. So I'm coming at this from a protein design and photosynthesis engineering perspective, but we could also imagine that natural ecosystems on exoplanets could evolve to have different types of photosystems working together. So we might not necessarily see a green bump and a red edge or something specific to a single photosynthetic pathway. We might see signatures of a mixture of different types of photosynthesis. Thanks.
Great. Thanks, Nate. That was really interesting. It tells us we need to be prepared to detect anything out there. So next we're going to talk about a good segue, photosynthesis in alien environments, and we'll have Nicoletta describing Earth organism survival under different stellar spectra. Go ahead, Nicoletta.
Okay, thank you. So this slide, just to recall the different light spectra irradiated and produced by the different stars. You remember that most of the potentially habitable exoplanets are orbiting red dwarf or K stars. This type of star has light spectra much different than our sun. Our sun is a G-type of star. You can see the light spectrum in orange, while the K stars are purple and in this picture, and the M stars are shown in green and pink. As you can see, the M stars represent the worst case scenario because they are depleted in the visible photons and reach the infrared. And this is challenging for the photosynthesis because oxygenic photosynthetic organisms absorb visible light. Next slide. And so to evaluate the potential of oxygenic photosynthesis under these light spectra, Riches and Lammer and co-worker consider Earth oxygenic phototrophs survival under dwarf light, and they did theoretical estimation of absorbed photosynthetically active radiation to evaluate this. But also laboratory simulation have been done so far, and a starlight simulator that is capable to reproduce the M-starlight quite in a reliable way was coupled with an atmosphere simulator chamber that can simulate the Archean atmosphere. And in this type of chamber, cyanobacteria or other organisms can be grown under the different light spectra. And it has been evaluated the responses, the photosynthetic and growth responses and plastic responses of the photosynthetic apparatus under this condition. And as you can see in the upper part of the slide, in the absorbance spectra, different type of cyanobacteria having different antenna pigments and capability to utilize infrared light or incapability to utilize it are all capable to manage to use the dwarf light and with the higher plasticity of their photosynthetic apparatus use this light. And in the lower part of the slide, you can see the oxygen evolution that has been measured under dwarf starlight and respect with red light and solar light. And under dwarf light, we can observe release, good release, efficient release in oxygen. So an efficient oxygenic photosynthesis, much higher than just when the cyanobacteria are exposed to red light and closer to the oxygen evolution obtained under solar light. So Earth's phototrophs can survive and make an efficient photosynthesis under dwarf light, at least cyanobacteria.
Great, thank you, Nicoletta. So now Nancy's going to provide a worked example of photosynthesis adapted to other planetary environments.
Thanks, Nikki. So Nicoletta showed us Earth organisms that are confined to special far-red or near-infrared light niches on Earth that could do well on the surface of planets orbiting red dwarf stars. So I'm going to work through an example, instead of for cyanobacteria, an example of an early photograph of purple bacterium and how that might be detectable on another planet, just to get you guys thinking about other planetary environments. So this is an example from Metz et al. What they did was take the spectrum, the absorbance spectrum of a purple bacterium, Blastochlorus viridis, which is known to absorb the longest wavelength for photosynthetic organisms, out to 1016 to 1040 nanometers. And that is about the 1000 nanometers, about where a certain kind of red dwarf star has some peaks in its photon fluxes. So the idea is that, okay, this organism could take advantage of that near infrared light. So Metz et al. Simulated the climatology of an aqua planet orbiting this red dwarf star. So this makes it kind of a simple planetary surface, but this is a tidally locked planet. So only one side of the planet always faces its star. And on the top here, you can see what happens when you have no bacteria on the surface. So this is the reflectance or the albedo visible in the top and in the near infrared on the bottom right. So with the bacteria, there's greater absorbance, there's actually less absorbance visible, but there's great absorbance in near infrared. And if you were trying to look at that, so you won't see this kind of detail with this telescope, we'll see a dot, but if you were to resolve this planet with, say, the Habitable Worlds Observatory, or the LUVOIR B, which was a predecessor concept mission to the Habitable Worlds Observatory in direct imaging, and to simulate the noise that you would get from this planet and it could tell if you had such a long certain integration time and want a signal to noise ratio of about 5, then you could detect some signal of the bacterial chlorophyll B at around the 1015 nanometer wavelength. So this is a worked example of a planet in a very different configuration and with an Archean-like atmosphere and oxygenic. So the big conundrum with Archean is that we haven't identified a really good atmospheric signal for life, though we know it existed then. So looking for a surface signal would be very valuable. And I just want to call out, of course, the parent star is not the only thing that would affect photosynthesis on the planet, but there are many other features. We showed a tidally locked planet with how the light would be concentrated on one side of the planet, so that's where the organisms would be. But all these other features, including atmospheric pressure, that can affect the conductance of the gases too. So these are all things to consider. And I'll give that back to you now, Nikki.
Great, thank you. So in summary, I know this has been probably a fire hose of information, but hopefully you can download the slide deck and come back to it for reference. But we heard from Nancy at the beginning, sort of the scope of the workshop, and heard about biosignatures that you may be a little bit more familiar with. So the vegetation red edge and oxygen buildup in the atmosphere from oxygenic photosynthesis. Then we learned a little bit more about light harvesting systems and diversity, electron acquisition and transfer, oxidizing water, which is a really key invention for the evolution of oxygenic photosynthesis, given that oxygen is a key biosignature, carbon fixation, photosynthesis in alternate environments, and then Nancy rounded it out with a great zoom out in looking at the planetary context. So next we're going to, if you want to advance the slide, we're going to transition to a fun activity that Nancy has created that's tied back to one of the earlier slides where she talked about the different scales of photosynthesis. And so Nancy, go ahead and describe this activity.
Okay. Hi. So last time we had a Moreau board where people had to slot the kind of biogenic or planetary feature that they wanted to observe into the right telescope technology. This time we have a Moreau board, and let me share my screen here. Oops, I'm gonna share a different screen now, and I see a lot of people have already dialed in there. So this one is just a linear configuration. Actually, is this show? Oh yes, okay. All right, everybody's in there already. So the idea for this is that you all have burning questions, and we don't know everybody's questions, but at every single scale of the way here, there is some kind of research question about what drive this level, the scale of mechanisms, and how that could impact what we can see of another planet. So what I'd like you to do is grab a Post-it note over on the side here. Oops, where are the Post-it notes here? Sticky notes on the menu on the left. You can pick a sticky note, and you're going to stick your question below the feature that you have of interest. And if you can figure out how to draw an arrow from your sticky note to that feature, that would be great too. It's a little bit, Miro has little control over connections between things. But go ahead and take about 10 minutes, 10, 15 minutes to generate questions like what do you want to know about these more that could affect the signal that we see from a telescope. Anything from the origins, how it evolved, how it would be configured differently, what the morphology would be, where would it be distributed on the planet, what kind of star, et cetera. So go ahead, post your questions. And for those of you who want also, the Miro board for the first primer is also open. If you want to also add there, you're welcome to work on both boards at the same time. So have at it, and we'll see what we come up with. Thank you. And any questions, and if you want to open your mic up and ask questions of people, and go ahead. Okay. Also, if you can, do put your name down at the bottom of your question. So we know who asked it.
Okay. Okay, I've got my mic on. Reverend Green. And something I've been wondering about is,
Oh, sorry, we cannot hear you very well. Maybe your headphone has the wires a little off. Can you change your microphone maybe, Bev? Or post it in the chat if your microphone's not working very well.
She might not be talking to us, so no.
Oh. Okay. Let's see what happens. Anybody in the other one? Let's see. You should have some music going. And if you can't see something, you can blow up the screen as much as you like. So you can expand it. For example, I'm blowing this up. Ooh, does retinol-based phototrophy produce significant spectral edge features that would be detectable? Thanks, Ali. Okay, if the red edge is the result of leaf structure rather than photosynthetic pigment, it's actually both. Okay, is it super contingent on the particulars of multicellular plants on Earth rather than being universal? Good question. So we'll probably look at a few more diverse organisms. Lichens have a red edge, but it's not very strong.
It's a good question. So cyanobacterial mats, microbial mats, actually do produce a fairly strong red edge feature, and it was in the one graphic that Nancy had showed in that Earth through time image. So it doesn't just require a 3D leaf structure. The trillions upon trillions of cells in a microbial mat are pretty effective at scattering light as well.
Okay, but it's not as strong as like a large plant tree canopy, though. If you stack leaves up on top of each other, near infrared reflectance really goes up.
No, they're actually, it's the same.
Well, that was my question.
Oh yeah. So yeah, I guess it's a matter of signal strength then.
So it seems to be just the fact that you have lots and lots of cells doing photosynthesis in one place that seems to cause that.
Yeah, I guess if you were to inject oil into, so this is the very early back in, I don't know, 50s or 60s, when they were looking at leaf spectra, they would inject oil into leaves to make the air spaces go away. And they, that way they can determine the index of refraction change that was causing the near infrared scattering. So I guess various configurations can alter it. Actually, Bev, what was your question? Let me take a see. Bev had a question.
Yeah, I'm just making conversation while we're waiting for that. So I've done control measurements where I've done plant leaves, and I've flooded the plant leaves and measured them alongside the cyanobacterial mats. And so it is roughly the same scale of magnitude of that, the steep increase in reflectivity in the red. But what are some other questions that are being asked?
So Bev Green asks, nobody has talked about what chemicals have been produced under conditions thought to be typical of early Earth, like amino acids. So Bev, are you meaning like prebiotic chemicals? Oops, maybe use the chat if your microphone's not working. Oh yeah, Beverly, your microphone's not working. Okay. But both, you can go ahead and post a note if you think something is missing that we didn't cover. I don't know if amino acids alone can be a sign of life, since of course they arise abiotically, but could be interesting to consider. Okay. I'll call some other questions here. Based on the planet's color, is it possible to design a hypothetical photosynthetic pigment protein that matches it? And would it also be possible to test whether such a photosynthetic pigment protein could actually work in reality? I think we've got a bunch of protein design people who think they can do that, but then we have to satisfy the criteria of viability, durability, or whatever criteria. There's some other, are alternate OECs possible? That's the golden question, Michael. Thank you for asking that. How can we prolong stable charge separation? Thank you, Colin. And Colin, that's just to promote efficiency to avoid back reactions. Is that why?
Right. Yeah. Essentially, just trying to stabilize holes and separated electrons for as long as possible, especially in a scenario like these red dwarfs where we have less light coming in on average, where it might not be feasible to simply scale up the antenna indefinitely.
How does the green sulfur bacteria do it at 1 photon per 8 hours? Or does it need a long charge separation, or does it just nab the electron quickly? Okay, something we discuss at the workshop some more. Any other cool questions? See, how might different metal concentrations impact the evolution of electron transfer proteins? From Simon. Thank you, Simon. Okay. So Ting Zhang wants to know what's Nate, for your alternative Z schemes. What are the spectral signatures that we would get? I guess we need to simulate those, huh? Or how do you calculate those?
Yeah, I guess it could be just about anything. So I don't know.
Okay, well, we'll have to fit into the golden triangle, or see what. Could it arise naturally and be efficient and be able to proliferate, reproduce?
These are some fabulous questions that you guys are asking, and it's this workshop obviously is very timely, and so for the Habitable Worlds Observatory, certainly want to design the most capable observatory to search for evidence of life. That includes both atmospheric biosignatures, as well as it will be capable of hopefully detecting surface biosignatures. But the point is that we can't just optimize it to look for the vegetation red edge or cyanobacterial red edge. I think one of the take-home messages from this primer, and hopefully from the workshop, is that we need to be able to detect pigment reflectance signatures probably anywhere in the spectrum, up to the theoretical limit. Unfortunately, you won't be able to see the surface of the planet in the blue, but we need to be prepared to hopefully see reflectance features out to, whatever, 1200 nanometers.
Let's see, how well could a leaf limit reflection of near-infrared light at the leaf air boundary to improve near-infrared absorption? I think that's a question for Nicoletta to answer. Thanks, Nate. There's one. Ting Zhang. Why didn't
sorry, I was trying to play with it. Yeah, sorry. Can you repeat the question, because I was trying to manage the mirror, and I'm not so familiar.
Okay, how well could a leaf limit reflection of near-infrared light at the leaf air boundary to improve? How well could it do it to improve near-infrared absorption? So, what's the marginal improvement?
Of the near-infrared reflectance
or absorption? Because your leaf epidermal cells your leaf epidermal cells, the conical shapes, are promoting near-infrared.
I really don't have an answer to that, because I don't know if there are plants that are able to reduce the reflectance in the near-infrared and utilize maybe some light in this case. I don't know if it is the question. I don't know if I got the point.
Okay, because your slide was about how these morphological changes were promoting near-infrared absorbance, right?
Yeah.
So you're measuring promotion of near-infrared absorption. So I think, Nate, were you asking?
Yes, but this is maybe due to the superstacks or the order thylakoids. The absorption of the cells that are conic in the epidermis of the leaves can just intercept light coming, very low light coming from different directions. As they are full of water, they can act as a lens and focus the light towards the lower tissue, photosynthetic tissue. And inside the photosynthetic tissue in the chloroplast, the superstacks or ordered membranes can enhance the scattering of long wavelengths photons and enhance the possibility of these photons to be trapped by a photosystem and so to photosynthesize and to evolve oxygen.
Thank you, Nicoletta. Okay, Ting Zheng is asking, why didn't plants on Earth evolve to use near-infrared? Is it due to strong atmospheric absorption in near-infrared region? Well, no, there's quite a lot of near-infrared coming down to the surface of our Earth. So the long wavelength cutoff is a major question for astrobiologists and just for photosynthesis. We did show, Nicoletta did show there are quite a lot of plants and algae that have been able to tune their chlorophyll A to absorb in the near infrared. And you still get oxygen evolution even out to 800 nanometers. It's a matter of efficiency. Also, Chris showed the chlorophyll D and chlorophyll F organisms. Those are only in cyanobacteria. They're not in plants. But they're definitely trying to eke out a living with any more photons they can that are available. So I think I tried to maybe hypothesize, well, the Earth is going to be redder in the future as these organisms continue to get more efficient using more of the light more of the solar spectrum. And our Earth is just not is not at the optimum yet, or is not using everything it can. So maybe we're going to have to be prepared to see an Earth in the future for the exoplanets. All right, we're going to give you another couple minutes and zoom out here. Thank you. These are great questions. Do put your last name too, so we can identify you and credit you for these questions if you can. Andrew R. asks, are macrostructures like leaves and plants possible for non-oxygenic photosynthetic organisms? And if so, why didn't they evolve on Earth? I think it's like a energy issue, and the I don't there's a body of work looking at what promotes what allows multicellularity in organisms. So I'm not an expert on that literature, but that's something we should maybe bring up more at the workshop. Definitely need some kind of different cellular structure. Wow, this is amazing. Okay. Oh, people asking things at the planetary scale too. Thank you.
Yeah, Tony made a good comment in the chat to answer that question. So in terms of like evolution of, anoxygenic photosynthesis on the early Earth, as Bob was talking about, yes, it uses other reductants, but those were limiting in availability, the volcanogenic reductants. And so that was an evolutionary selective pressure that drove the ability to be able to oxidize water, which as Tony points out is everywhere. So therefore oxygenic photosynthesis wins out.
Great, thanks, Nikki. Yeah, water is available widely, much more than the anoxygenic reductants. So, okay. All right, let's give one more minute. Then we'll have a closing. This is great. Wow, if you guys can go back to the exoplanet one too and throw in a few more ideas, that'd be great.
Cool.
Let's resume. Let me see if the other memory boards, anybody looking at that one.
Thank you guys for being so engaged. This
is really fun. Okay. All right. Any other good ones? Let's see. Oh, Stephen Moses asks, why did life choose chlorophyll-like structures as opposed to PAHs, iron sulfide complexes, keto acids, melanins, etc.? Okay, so at the end of our slides, with the slides we post, we'll have some slides of literature. These papers are far and few between, but every 10 years somebody comes out of the paper, why chlorophyll? And what's the advantage of chlorophyll? So we'll post those literature on the slides that we upload. But the early ones were like 1970s, David Mazeral, and then another one in chlorophyll A in particular. And the Bjorn et al. Came up with a new one again much later. But these pondering this is a long process for science to understand.
Yeah, in terms of the question about the pigments in prebiotic chemical, I just missed it. I have talked to our astrochemists at NASA Ames. These are folks that characterize like PAWs in the interstellar medium. And I asked them, like, do you ever see like a protopigment type structure out there? And I think the answer is that, well, number one, those spectra are so complex to disentangle, but maybe could be some protopigments that look somewhat like a paw out there.
Okay, we are at a little 9 minutes before the end of our period, so I'm going to close this sharing, and we'll go back to summarizing and going forward, seeing what we're going to do next. So thank you everybody for adding to this board. I think we'll probably print it out as a poster for the main workshop, and thank you. Maybe people keep adding to, you can feel free to keep adding to it as the thought comes to you, and we can maybe call out these questions at the workshop to discuss further. Okay, I'm going to go back to sharing the the Google Slides. All right. Thank you. All right, just to wrap up. Okay, in the middle of looking at all these different kinds of possible signs of life, I just want to rehash what we went over at the first primer, that for all of these, you have to be aware that environmental context, and be wary of false positives and negatives for signs of life. And there have been a few frameworks posed to make sure all the people from different scientific disciplines, are checking each other and have a system for being very careful, because when we announce there's evidence of sign of life or something novel, you really have to have extraordinary support for extraordinary claims. So here's a few systems for coming with standards of evidence, and also for constraining the confidence with which you announce your possible detection of life. So I encourage you to look up these papers for just honing in on how to constrain their confidence in detecting life. Also, there are a few resources online for getting spectra of different pigments and organisms. So these are just a smattering of things you can look up. There's much more of this on the web, but these are downloadable data sets you could use to play with ideas. And there, just to call out some of the questions, and I hope you understand why we're asking: can there be alternative primary photopigments to chlorophylls? Is there a long wavelength limit? Is there a limit to the number of photosystems that can be strung in series to support electron transfers? These are all questions we still have to resolve. Could rhodopsin-like proton pumping support autotrophy? And what are some alternative methods from artificial photosynthesis that can maybe arise naturally? Or maybe they'll serve as creating technosignatures for an advanced planet. And of course, how did the oxygen evolving complex arise? We don't have actually any good sense of precursors for it. There's some ideas that manganese oxidation could have been some kind of precursor, but still a lot of research to do there. One of the things people want to know is, because of this inefficiency of Rubisco, are there alternatives? And what's the ultimate efficiency for carbon fixation? And as the planet ages, its CO2 will be drawn down by weathering in the carbonate silicate cycle. So how long could photosynthesis be viable over the life of a planet? And it depends not only on CO2, but also oxygen. And what are the spectral signatures? So thank you, everyone, for joining us. Nikki, would you like to go ahead and tell us about the next stage?
I was gonna say, keep going. No, we're just, thank you so much for joining us today. You guys were fantastic. It was great to see all of your questions and the Miro board. And next step then is our in-person workshop, May 15th through the 17th, just preceding AppSciCon. So we look forward to seeing you there and continuing discussions.
I guess we're 5 minutes early. People can continue to play the board. There's a few other slides you didn't show, but there's some literature we'll throw out. These are some critical papers you might want to look up. What are properties of chlorophylls that make them special? And then there's some, there's a little bit of summary. And there's some papers that also, they're always they're like 10 years apart usually, about what about photosystems that could be optimized for other stars. So you can look these up there. These are all modeling theoretical studies. Takizawa has an interesting paper playing on Nate's idea where two photosystem apparatus could have the PS2 analog and the PS1 analog absorbing in different spectral ranges. And how does that happen if the organism evolves first underwater, the photosystem evolves underwater, where it's the red light gets absorbed, and then it comes to the surface. All of a sudden, the spectrum changes at the surface. So these are interesting papers to speculate about. So thank you, everyone. And anybody have any questions? All right, Bev, did you get your microphone working? What your question? Did you get to post it? Did you get to post it in the board?
No, I didn't. I couldn't actually see the board properly.
Oh, I'm sorry. Okay, you'd be posted in the chat. We'll post it for you.
I'm just looking to see if I've read all the.
All right. Thank you. Thank you, everyone, for joining. We're just going to stay on. If anybody wants to chat some more.
Nancy, I'm going to step out. I have to get ready for my next meeting.
Okay. All right. Thank you. Thank you, Nikki.
Thanks, everybody. Bye.
All right.
Bye-bye.
Thank you all. Thank you. All right. Sorry, I ended a little. I need to let people go.
Bye-bye.
Okay, bye. Oh, Elizabeth, that's you. Yay, good to see you.
Bye.
Okay, great.
See you.
Thank you. Okay. Okay, this is just us now. Okay, there are a few others. Okay, Gary, Nate, Chris, Alessandra, thank you for running. That was awesome. Oh, Bill, you made it on. Okay, you got successfully on. Thank you for joining.
Thank you, Nancy. Sorry for being such a pain.
No, not at all. Okay, yeah, that was really well tailored, Nate and Gary, for the artificial physicists to astrobiology. Thank you.
Thank you.
Thank you, everyone, and thank you, Nancy, for that wonderful introduction, comprehensive introduction in 5 minutes of everything.
Okay, hope it wasn't too rushed. Thank you. And all right, I guess see you in May in Wisconsin.
Yeah.
And we're just I guess we're just going to coordinate the bus and what else? Oh yeah, we're going to post the program on the website. We just yeah, we revised the program a little bit to get some of the orders and get some of the fundamental stuff earlier in the talks earlier, so we can set up our workshop, our working group topics earlier. So we'll be posting the program hopefully by Friday. We'll see. Okay. Thanks everyone. And all right, Alessandro, it over there. Gary, that was great. Thank you so much.
Oh, thank you. Yeah, thank you.
That was really great.
Really enjoyable time. Thanks again.
Yeah, it was enjoyable. Thanks. Gaojong, nice to see you. See you in Wisconsin. And Dongsuk, why are you dialing in fun? Okay. All right. Thank you, everyone. I'm stopping the recording. Recording. All right.